Wine. Have you ever stopped to think how long it's been around? More than 7,400 years. A long time, right? This has been achieved by means of a very simple common denominator, the Earth. And from it, our dream is born. We're pleased to present WineSeq. WineSeq is a veritable revolution that helps wine growers all over the world better understand their surroundings. From now on, we can find out the real status of our vineyard and its fruit, discover what exactly to add to each terroir, minimize risks by anticipating potential diseases in the plant, and even enhance the quality of the must to produce outstanding wines. How? Thanks to a new technology developed by Biome Makers, we can now read the microorganisms in the soil of the vineyard. And you'll be amazed at how much the soil has to tell us. The procedure is simple. All we need is a sample of soil, of grape, or of wine, which you send to our labs. Then, using a simple app, you'll be able to read the results. Once you're a member of the WineSeq Club, you'll have access to precise information on your soil. You'll be able to access our community and make the best decisions at any given time to help you start making the wine of your dreams.
Okay, let me introduce myself. Adrian Ferre. I'm one of the co-founders of Biome Makers. We are really happy to stay here and come here because we received the invitation from Vinidea to come and pitch about this topic, which is quite hot right now. Our company was founded in San Francisco, and last year we came to Europe and we moved all the R&D activity. And what we know about is about DNA sequencing technologies and cloud computing and machine learning tools. That sounds familiar, but the combination of these two technologies is very interesting. It has been widely applied to health, to human health, but nobody realized that we could also use this technology to provide value to a very interesting field as the winemaking. Because, honestly talking, we don't know what's happening, almost nothing about what's happening at microbial level during the fermentation process, during the winemaking process. We don't know much about the origin. And, well, if we are able to benefit from these technologies, that now is very common to hear new companies, new startups working in ag-tech, especially everybody knows about the drones, all the information management systems that combines different layers of information and then try to provide value out of mining those data. So the approach that we decide to apply here is a bit different. We combine these technologies, but we also deliver a completely new layer of information, which is the full microbial profile. So all the bacteria, all the fungal species that we can find in any environmental sample. And this has a very interesting use for us because usually when we think about the connection between the vineyard and the cellar, usually we think about the grape. But there's really something else because regionality in wines is very important. And that's how we started thinking about this technology. And the first thing we did was to review all the scientific literature and realize that there is over 2,000 different publications, and we didn't know what's the information included in that. So the first work we did was to, just let me grab the, was to collect all this information and put it in a common platform, common language that everybody can understand it. So we have the Wikibiome, including all the species that have any relevance on winemaking or plant health. And this is free. Anybody can access to this information. And you just need to go to register to the wineseq.com, click in sign up. And we also have a community where all the researchers that are already in our system and all the winemakers can make anonymous questions. And all the community can reply those questions. So I invite you all to create an account and place any question that you have related to your business or out of curiosity, because it's going to be very interesting to know the replies from the people. And don't forget to place the Enoforum tag, because that's the way we know that this is coming from this event. But today, I will say I don't want to be the star, so we have a very nice panelist, and I want them to introduce themselves because they are the really ones who can provide a nice view on the influence of the microbiome and all the approaches and new novelties that we can find then.
Buongiorno, so I'm Stephane Compant, I'm just on the picture here, and I'm a researcher working in the Austrian Institute of Technology in Thun, in Austria, and I'm focusing mainly on plant microbe interaction, how the microbes that they are detrimental for the vine or beneficial for the vine, how they interact with the plants, what they are doing with the plants, where we can find them, and to understand how it's work. Because when we see, for example, grapevine plants, like other kind of plants, they are interacting with a plethora of microorganisms, viruses, bacteria, fungi, or mycetes. And it could be in a positive interaction way or also a negative way. But we need to understand better the superorganism, how it interacts with the microbe, because the vine plants are not sterile system. And these plants can have different kind of microbe inside root, inside the trunk, for example, trunk pathogens, on the surfaces of leaves, on the surfaces of flowers, berries, but also inside different plant organs. So I try to understand to what kind of community, bacterial and fungal community is associated with these grapevine plants.
I'm Gobbi Alex. I'm an Italian PhD student currently involved in a micro-wine project in Denmark. I'm working for Aarhus University. And so I'm going to spend, since I'm here as a representative of this micro-wine project, I'm going to spend a couple of words about it. It's a big international European network financed by Marie Curie that involved 13 PhD students like me, and no, 15 PhD students like me in 13 different universities around the world. And the aim of this project is to enlighten what is the effect of the microbial community on all the wine production process. And we are doing this applying the newest technologies that are currently available on the market. And in our sub-project, we are trying to study this effect under many different aspects that goes from the vineyard to what is happening in the winery during the fermentation. And hopefully we can provide some answers to unanswered questions that are like until now. And yeah, that's it.
Hello everybody, my name is Alberto Acedo. I am the co-founder, the other co-founder of Biome Makers and the technical director. I am PhD in genomics, specialized in precision medicine. 5 years ago, we decided to start a company to analyze the genome of the people to prevent hereditary disease, establish a risk, for example, in breast cancer or in colon cancer. But 2 years ago, we decided to move with Adrian to San Francisco to start a new idea. Replicate the same concept like precision medicine in other fields, in agriculture. And now we develop WineSeq. WineSeq is a system to use genomic technology and collect different metadata associated with the location, kind of soil, kind of weather, and create algorithms using different big data systems to predict risk associated with the vineyard, prevent plant disease, and for the other hand, use this information to improve the quality of the wines. Because with WineSeq, we can analyze all kinds of samples. We can follow the microbiome community from the vineyard to the bottle and correlate this with flavor information or other kind of information.
So thank you very much. Another thing is that we love if you use your mobile phones. There you have all the Twitter accounts. Feel free to tweet anything that you want. So let's go into the topic, which is the microbiome. We hear microbiome all the time. It's a very trendy thing. What is that? So Stephane, can you give us some light about what's the microbiome and the influence in the plants?
Yeah, it's a good question because we listen more and more about microbiome. What is a microbiome? In fact, what I told you just before, if you see the plants is not a sterile system, and it's interact with a community of microbes, and this community of microbes associated with the plants, it's what we call the microbiome. So it's all the community of every kind of microbes in different plant parts interacting with the plants. And when you have, for example, grapevine plants growing in some region, they have some specific microbiome, but they have also the same kind of microbiome that the same cultivar in another region. But there is also modulation of this. So it's just the community of microbes interacting with the plants, what we called before the microbiota. And it contains also all these microbes, they interact with the plants, and we have a super organism with a collection of genome of the plants, but also of the microbes. So there is functionality of this microbe associated with root or the higher part of grapevine plants.
Thank you. So then I have a question for Alex. Is there any relation between the microbiome and terroir in broad sense?
Okay, thanks for the question. Actually, we know that microbes are like the main actor in many different steps during the wine production process. So, and we also know that there is a correlation between the distribution of these microbes and the geography and the places in which the grape wine grows, and also in which the grape are processed, like inside the wineries. So for sure there is an interaction between the microbes and the wine, and this interaction can be due to many different factors. It can be geography, but it also can be how geography, but also the human, how the human affect the production process. And I would say that we know something about the interaction, something that we can measure based on the aromatic profile of the wine. And this happens when we did an analysis on the strain of the famous Saccharomyces cerevisiae. This was made, for example, in New Zealand a few years ago, in which they were correlating these strains of Saccharomyces coming from different local places in New Zealand, and then they were correlating with the aromatic profiling of the wine. So in this way, they were demonstrated terroir, at least the microbial part of the terroir, not only exists, but is also active in doing something to make the wine unique, how it should be.
Good, so that means that whenever we hear about terroir, the climate influence, the practices, everything could be related to microbiome. So, Alberto, which kind of information can we get from the microbiome? That's probably the question.
For my perspective, when we talk about microbiome, we talk as first an indicator, because in the microbiome population, we can find pathogens related with different diseases in vines, but we can detect other positive species, like plant growth promotion species, or, for example, fixing nitrogen bacteria. And all these species are related with the health of the vine. And the health of the vine is related for the quality of the grape. For the other hand, looking at the soil, we can detect, Saccharomyces species or Enococcus species, but we can detect all kinds of species related with the fermentation process. If I ask you about if you know all the species related with the wine fermentation process, probably you mentioned Saccharomyces, Enococcus, Acetobacter, Lactobacillus, Pediococcus. But when we analyze wine samples, we can detect more than 100 different species in different moments of the fermentation process. It's a evolution of the community, of the microbial community, to survive in a high or difficult environment. For the other hand, when we look at the microbiome, we can use the microbiome as a biomarker for wine problems. We can detect, for example, Brettanomyces in soil samples, in grape samples, or in wine samples. And if we detect Brettanomyces in grape or soil samples, we can prevent the effect of Brettanomyces in wine. But for the other hand, as Alex mentioned, we can correlate specific signatures in the microbiome related with the agriculture practices. Now we have an internal research project to try to find specific markers, specific species that appear only in ecological vineyards and other species that appear always in conventional vineyards. For the other hand, we can use the microbiome as a biomarker of the terroir. You know that terroir is a concept. Terroir is a mix of tradition, it's a mix of soil, a kind of soil, weather, but all these factors impact in the microbiome profile in the soil. And we can detect the origin of the microbiomes according with the finger prime, the abundance of each species and the different species we find. And we can compare the species we find in a specific vineyard from Italy compared to Spain and see what are the differences at the microbiome level. And for the other hand, probably we can follow the effect of climate change, because if the temperature of the air is increasing, for example, in the Mediterranean countries, in soil can increase to the temperature. And we can measure the effect of the microbiome profile, for example, related with the survival of the plants, of the health of the plants.
Well, panelists, please feel free to interact and contribute as you wish. But that's a very interesting point, talking about climate change. Well, especially for Southern European countries, that we really see that other countries as Denmark or UK are starting to have their own wine. We are having a really hard time because hydration, because well, there is less nutrients. So we see a lot of effects going on because of the climate change. So, Stephane, is there any relation between also the diseases, the prevalence of the diseases, the climate change, and is the microbiome involved somehow?
Very nice question, yeah. In fact, with the climate changing force, drought stress, high temperature condition, we have the risk to have more disease. And there is a rupture in the equilibrium of the microflora, and we can have also increased carbon dioxide. We know, for example, if we have increase of carbon dioxide, high temperature condition, we can get more downy mildew. If we have more high temperature condition, we can get also more grape trunk disease, such as ESCA, but also Phomopsis dieback and some other. In fact, the microbiome of plants is depending of the plants, it interact with the plants, but it's also subject to environmental factors. And this microbiome can be modulated by environmental factors. And if there is not the same equilibrium and there is a dynamic of this microbiome, it can lead to change, to have more pathogens, but also to make emerging pathogens, or that some good part of the microbiome, the natural biocontrol agent, for example, interacting with the plants, will not work like before because of the high temperature condition, and we will have a switch in the community of microbes that can lead to more kind of a specific disease or reduce some other.
Yeah, Stephane, I agree with you related with the effect of temperature or other things in the microbiome profile, because microbiome in the roots is like our microbiome in our gut. It's essential for the plant, it's essential for the nutrition. But why, for example, we are looking for, we can see now in the vineyards, for example, in Spain, more incidence of trunk disease. Is this the same species or the same strains, or we can show changes in the species? The question is always when we analyze the soil, we can find, for example, pathogens, but we can detect symptomatology in the plant. But if the weather conditions change, we can start visualizing the symptoms. But now, do you know if are the same strains or are different strains to affect the plants?
It's an evolution, and also what we have, you know, when we see grape trunk disease, we have only one pathogen. We have several pathogens, and what we need to know also the microbe associated with the pathogenicity related to grape trunk disease. They are living in symbiosis with grape plants up to 7 or 10 years before that the disease arrive. And this disease can arrive with a plethora of things together: the microbes, the environmental condition, also a cultural practice, with running in the field, and there is also more high temperature condition, and there is a lot of different kind of trunk disease, and we have no solution now, but we are working on the interaction of this microbe with the plants and see that the concept of one microbes and one disease is not true for this case. And we also expect that there is more than we know about the number of fungi, but there is also a possibility of other kind of microbes preparing the place for this pathogenic fungi. And there is some current biocontrol solution for this to prevent, but not curative, of course. Yeah. So you should live with your grape trunk disease, sorry. But I know that it's pain.
Yeah, so climate change is actually a big threat for all the Southern European countries, but in the Northern European part, they are quite happy about it, so they would like to get some warmer weather in order to, like, in somehow improve the cultivation of the grape wine. And what I was saying before about the terroir and the fact that it's unique, it doesn't necessarily mean that it's always the same. And climate change can affect the microbial community, and in this way, maybe the terroir also can change. It might stay unique, but simply be different. And I guess this is why it's important to keep it monitored along the years, because at least for my preliminary results that I had, like obtained in Denmark, I could see that in 2 consecutive years in the same field, there is a shift in the microbial community, and this is probably due to the weather condition that has been changed a lot in the last 2 years. Probably it was just random association, but this has been changed, and I could see this change in the microbial community. And what I wonder actually, and I want to ask Stephane, is it like this shift in somehow can be an indicator of the evolution toward a condition of pathogenicity inside the vineyard? So can you think that in the future we will be able to give a meaning to this shift and be able to predict maybe the appearance of some pathogen?